Hello, everyone. Welcome back to my Cellchat video tutorial. In this video, I'm going to show you how to perform systematic analysis of cell-cell communication networks. So we can load the required packages. So in my previous video, we analyzed the dataset for injured human skin. So we can load the dataset. So for today's analysis, we are going to analyze all the signaling pathways. So before we do the analysis, we can have a look at the dataset. You can see we saved the signaling pathway analysis in the NetP data slot. At the moment, in the NetP data slot, we have 47 identified signaling pathways and also the probability for each signaling pathways in 12 different cell types. So we will see all the analysis in today's video in the NetP data slot. So first we can calculate the network centrality scores. After the calculation, we can have a look at the data. If we look at the data now, you can see in the NetP data slot, we added the centrality score into the NetP data slot. We have the calculation for 47 signaling pathways. We can use one of the signaling pathway, for example, the TNF signaling pathway as the example to show the calculation. So you can see the analysis calculated the outer degree unweighted, in degree unweighted, and also the outer degree and the in degree. It also calculated the betweenness to show the cell types that mediate or influence the signaling pathways. So we have the calculation. Then we can use the scatter plot to visualize aggregated communication network for each cell type. If we run the scatter plot, we can see the aggregated signaling pathways for each cell type. Let's zoom in. You can see we have 12 cell types here. The x-axis shows the outgoing interaction strength for each cell type, and the y-axis shows incoming interaction strength for each cell type. And the circle size indicates the total count in each cell type for both the incoming and outgoing interactions. So this is the aggregated signaling pathways for each cell type. If you want to see individual signaling pathways in each cell type, we can also use the scatter plot to visualize the signaling pathways that you are interested in. So for example, we can use the scatter plot to plot the TNF signaling pathway in different cell types. So let's zoom in again. You can see now for the outgoing interaction strength, that's the cell types express TNF ligand. So we know from previous analysis, T cells, C cells, and dendritic cells express high level TNF. And you can see the strong incoming interaction strength for the fibroblast because the fibroblast express the TNF receptors. So this is the scatter plot for TNF signaling pathway. If you want to plot more than one signaling pathway, for example, here we can use the concatenate function to plot a CXCL pathway and a CCL signaling pathway in the same scatter plot. We can zoom in again. You can see now It is the aggregated incoming and outgoing interaction for both CXCL and CCL pathway in different cell types. So this is the scatter plot. We can also use the heat map to visualize the cell types that express high-level ligands or high-level receptors. If we set the pattern as outgoing, so we will plot the expression of ligand in each cell type. Let's run this code, and we can zoom in. You can see now, so we have 47 signaling pathways listed on the left-hand side, and 12 cell types listed on the bottom side. So the dark green color indicate high level of expression for a ligand expressed in different cell types. So on the right-hand side, you can see the contribution of each signaling pathway to the overall communication networks. You can see from the outgoing signaling patterns, the LC cell type contributed the most signaling pathways to the whole communication network. So this is the outgoing signaling pattern. If we set the pattern as incoming, then we can plot the receptor expression in different cell types. Let's zoom in again. You can see now it is the plot show the incoming signaling patterns. The dark blue color will show the cell type that express high level the receptors for each ligand. Once again, we use the heat map plot and plot the both incoming and outgoing signaling patterns for all the signaling pathways. If you are only interested in some signaling pathways, you can just plot the signaling pathways that you are interested. To plot the signaling pathways that you are interested, we just need to add the signaling pathways into the heat map plot function. Once again, we can use the CXCL and CCL signaling pathways as an example to show the heat map plot. If we set the pattern as outgoing, that means the cells express high-level CXCL and CCL ligand. We can zoom in. You can see now we only have two signaling pathways, CXCL and CCL. You can see the ApoE positive fibroblast express high-level CXCL and NK T cells express high-level CCL. If we set the pattern as incoming, then we will plot the receptor for both signaling pathways. We can zoom in again. You can see now the ERC cell type express high level for the CXCL receptors, and the NKT cell express high level the CCL receptors. So from the ligand and the receptor expression, you can see the CXCL signaling pathways is a paracrine signaling pathway, but the CCL signaling pathways is a autocrine signaling pathway on the NKT cell types. So far we plotted the ligand and the receptor separately. We can also use the signaling role network function to plot the individual signaling pathways, but we will see the expression of both ligand and the receptor. This function also reveals the cell types that could mediate the signaling pathways or influence the signaling pathways. So we can use the TNF signaling pathway as an example to show the TNF signaling pathway between different cell types in this data set. So let's zoom in. You can see the center will be the cells that express TNF and the receiver will be the cell types that express the receptor. From our analysis, we know TNF was secreted by the immune cell types and their receptors are expressed on the fibroblast. So immune cells are the major cell type to mediate the TNF signaling pathways. And the fibroblast that express high-level TNF receptor, immune cells that express high-level TNF ligand are the influencer defined by the Cellchat analysis. So you can see through the centrality score analysis, the data can show the dominant cell type that express the ligand for each signaling pathway and also the dominant cell type that express the receptors. So after the centrality score analysis, we can identify the global communication patterns. Once we identify the global communication patterns, we can explore how multiple cell types and the signaling pathways coordinate together. For this data set, we only analyzed the secreted signaling pathways. So we are going to analyze the outgoing pattern and the incoming pattern separately. Cellchat uses the select key function to identify the communication patterns. This is the function from the AMF package. So first, let's use the select key function to determine the outgoing communication pattern for this dataset. Okay, it is done. Let's zoom in. So you can see select key uses two parameters. One is cophanetic, the other one is silhouette, to show the number of patterns up to 10. The default set for this function, the number of patterns is 5. If you have more than 5 patterns, it's more difficult to explain the data. So according to the online tutorial, the best number of patterns that we can select it from here for further analysis is the number that start to should dramatically decrease for the measure score. So you can see here, 3 is the best number of patterns for our analysis. So we can set the M patterns as 3 for the outgoing patterns. Then we can visualize identify the communication patterns. So let's clear the plot here. Then we can generate the new plot. Okay, let's zoom in. You can see, because we selected 3 patterns, so we have 3 patterns here, pattern 1, 2, and 3, and you can see the contribution for the cell types for each pattern. You can see for pattern 1, it's a fibroblast contribute to pattern 1 communication network, and for pattern 2, it's the NK cells and the T cells. So dendritic cells and the LC cells contribute to the communication network for pattern 3. So on the right hand side are the signaling pathways linked to each pattern. Because this is the outgoing communication pattern, so the red color indicates the cell types that express high-level ligands. So we identify the 3 patterns for the communication network in this dataset. We can also use the river plot to plot the outgoing communication patterns. Let's zoom in. You can see on the left-hand side are the river plot for cell types correlating to each pattern, and on the right-hand side are the signaling pathways correlating to each pattern. So in addition to the real plot, we also can use the dot plot to plot the outgoing communication patterns. Let's run the dot plot and zoom in again. You can see from the dot plot there are 3 patterns. On the top are the signaling pathways associated with fibroblast in pattern 1. In the middle are the pattern associated with dendritic cells and LC cells, and in the bottom are the signaling pathways are associated with T cells and NKT cells. So you can see from the dot plot, each signaling pathway is almost exclusive to one pattern. So this is the outgoing pattern. We can also run the same analysis for the incoming communication patterns. So first we run the select key function to identify the pattern numbers for incoming communication pattern. So you can see select key identify the pattern numbers for incoming communication signaling pathways. From both parameters, you can see 3 still is the best number of patterns for the incoming signaling pathways. So we can set the n patterns as 3 again. Then we identify the communication patterns for the cell types and also the signaling pathways. So let's clear the plot first. Then run the communication patterns for the incoming signaling pathways. We can zoom in. You can see again we have 3 patterns. Pattern 1 is associated with fibroblast. Pattern 2 is associated with LC and dendritic cells. And the pattern 3 is associated with T cells and NKT cells. Because here is the pattern for incoming communication networks. So on the right hand side should be the receptor expression for each ligand associated with 3 different patterns. So we perform the communication pattern analysis. If we have a look at the data set now, you can see in the NetP data slot, we added the pattern in the data slot. In the pattern data slot, we perform the outgoing patterns and also incoming patterns. So as I showed you for the outgoing communication patterns, we can also use the river plot to plot the incoming communication patterns. So let's zoom in. You can see the cell types associated with each pattern for the river plot. On the right hand side are the signaling pathways associated with incoming patterns. So in today's video tutorial, I'm going to show you one more analysis. That is to group the signaling pathways based on their functional or structural similarities. The functional similarities is easy for you to understand. That means if we group some signaling pathways together, signaling pathways in the same group probably have similar biological functions. So we can analyze the similarity, set the type as functional. So we can analyze the similarities for the signaling pathways in this dataset based on their biological functions. Then we can embed the functional similarity into the dataset and cluster the signaling pathways into different groups. So after the analysis, we can reorganize the signaling pathway groups based on their functional similarities. So let's run this function and we can zoom in. You can see we have 4 groups here. Based on the analysis, each signaling pathways in each group should have similar biological functions. We can also zoom in to each groups. Let's plot it again. You can see now we have 4 plots. We separate the signaling pathways into 4 groups. So here we group the cells based on their functional similarity. We can also group cells based on their structural similarities. The structural similarities is more difficult to understand in the beginning. I read their original publication. The structural similarity analysis is based on the ligand and receptor structure. For example, if the ligand receptor have multiple complex or not, or they can be grouped based on soluble agonist and antagonist, or based on the similarity of their stimulatory and inhibitory co-ligand and co-receptors. So let's analyze the structural similarities and then embed the data into the Cellchat object. Then we can cluster the signaling pathways into groups based on their structural similarity. So after the analysis, we can visualize the groups for the signaling pathways based on their structural similarities. So let's zoom in again. You can see, based on their structural similarities, all the signaling pathways have been grouped into 4 groups again. And once again, we can zoom in to see individual groups. So here I only show you how to do the analysis. If you really want to understand the biological means for different groups, you should read their original publications. This will help you to explain your own data. So we finished the functional and structural similarity analysis. Let's have a look at the Cellchat again. You can see in the NetP data slot, we added the centrality score analysis, the pattern analysis, and the similarity analysis. In the similarity data slot, you can see we have the functional similarity and the structural similarity. So let's save the data set. Then we can use this data set for my next video tutorial. Okay, that's it for today's video tutorial. I want to emphasize here, in this video tutorial, I only showed you how to perform the analysis. To understand the biological functions of those analysis and also to help you explain your own data, I suggest you to read the original publication for the Cellchat package. So I hope my video tutorial can help your data analysis. Please subscribe to my channel if you are new to my channel, and also share my videos with your friends. Thank you, and I hope to see you in my next video tutorial.